here on IODP Expedition 337, the Deep Coalbed Biosphere Expedition, and we are here to study microbial life that's associated with a deeply buried coal bed about 2 kilometers underneath the seafloor.
How deep life exists and what is the functioning of microbial life and how do they evolve during the history of Earth's change.
What we don't really understand are the limits of life. So we don't know really what the extension is of the deep biospheres. We think that temperature is regulating it, but there could be other factors such as energy or water availability or simply physical factors. Is there enough space? Because the pressure goes up when you go deeper and material gets more and more compacted. Microbes may just not find a very good habitat there anymore. And those are all issues that we don't understand. And to understand these, we really have to drill that deeply.
Our humans always consume some energy, like food. I think the deep subseafloor microbes also has need energy and to survive or for geologic time or long time. And if there is so many microbes, I think there's some unknown energy supply, which it might be a deeply buried coal bed.
This is the first time that we had the chance to study this particular situation, coal underneath the seafloor and microbes, and we are interested in finding out what the coal, to what degree it serves like as a substrate and source of energy for these microbes. What's unique about our program is that we drill very deep, and so one of the major challenges for us is to get pristine samples from these coal deposits and most likely also deeper. So we use a drilling technology that is widely used in petroleum industry, but this is not made for contamination-free drilling. So we have to devise new technologies and strategies to minimize this contamination. So we are walking a very fine line here, what technology has to offer and between our scientific demands.
Attention. No, goes in the sector.
There are multiple sources of potential contamination. The probably most drastic one is comes from the drilling, where drilling mud is circulated through the borehole, and this contains chemical additives, but also seawater, potentially microbes from the ocean, and they are introduced into small fractures in the drilling core. That's what we want to avoid.
Incredible.
To minimize contamination, we have a set of steps that we implement before we take samples. We scan cores for potential fractures. So my colleague, co-chief scientist Fumio Inagaki, and I have to make the decision after each core. We scan with an X-ray scanner these cores three-dimensionally. So I'm searching for potential places where drilling fluid can penetrate, and this would be an example where we have a crack, so that is probably happened through drilling, and we have this drilling fluid through the drilling technique, and that can penetrate in here. This So if we have this as part of a sample and analyze it for chemistry, then our results will be wrong. So we have to carefully check where we take samples.
Now we have IW sections. Please cut 18 centimeters from the top. I'm quite happy that we have a multidisciplinary expertise team. We have a geologist, sedimentologist, and physical property. And of course, this expedition is dedicated to microbiology and biochemistry. So we have a great staff of life science. And it's, there's some fruit inside, so you can, the entire you can use.
Okay,
and there's some 2 pieces inside.
We have about 30 scientists from different countries and different backgrounds, so it's the best part of my job to combine each other's interests together and it makes them work into a one goal. And I think this cruise has a very good science party team, so they are working very closely with different groups, so I think that is the best part of my job.
We're currently drilling through marine sediments, and diatoms and calcareous nanofossils live in the ocean. They're photosynthetic phytoplankton that float in the ocean and thrive off of nutrients in the water and of sunlight. So when they die, they fall and accumulate on the ocean floor. And that accumulation builds up over time, and now we're drilling through it. And it's been building for millions and millions of years. The first occurrences of species, the last occurrences of species are correlated with these other time proxies, and that's how micropaleontologists can use microfossils to date marine sediments. Our scientific objective says that we're drilling for coal that's Eocene age. So as I go through the samples, I'm looking to see what the age of the current sediments are, and then the co-chiefs are able to determine how far along we are in the drilling and how much farther we have to go. And they decide how they want to drill, if they want to core or if they want to get cuttings or how they want to proceed with drilling. What you're looking at right now is Neodenticula seminae. Its first occurrence appears approximately 2.5 million years ago, and this species is still living today in the oceans. This species is called Neodenticula koizumii, and it has a range of approximately 2 million years, between 4 million years and 2 million years old. Identified both Neodenticula koizumii and Neodenticula seminae in this sample, and based on the appearance of both of those species here, I can tell that this sample is between 2 and 2.5 million years old.
Any idea what the age could be?
I don't have any idea on the age right now, but I'm seeing more indications of a marine environment than a terrestrial environment.
Okay. Right now, for us, it's important whether we see the continental or the marine diatoms for our decision whether we continue drilling to take samples or whether we just drill down right now and move on and potentially don't recover the interface, marine terrestrial interface. So it's a difficult decision right now, and we also have split opinions. So we have to act fast. My vote would be follow the plan and drill deeper and take the core in 100 meters or something.
So Yamada-san also thinks that there is no definition based on seismic profile.
No, so not really sure. And what time we refer to continue after this.
So you mean there's a chance for lithologic change coming? Or
No, not really.
Not really.
Well, just one reflection on the profile, and that is 1800 meters. And I think the upper boundary of the sandstone might be that reflection surface.
Okay.
Shall we do it down to 70, 1820?
That was our original plan, yeah?
Yeah.
1820. Yeah.
So when I told them that we were looking at late Miocene sediments based on this sample, they could tell that they were far from their target. So they knew that if they drilled quickly, that they weren't going to miss their target of that coal bed. If we were closer to the target, then they would decide to core and recover good solid sediments.
次の泥岩がシェルを組んでたんで、テストやるのやる気がないから。リオラン。
Right, no real. Let's go for coal.
We now made a consensus.
Consensus.
Consensus. See, we will do it down to 1820. We will not take another core. We do it down because of the time constraint, and there is no guarantee to see the lithological change if we take another core. So there are so many, some constraint for realizing this work for the successful.
We race against time. In this role as a chief scientist, we carry also responsibility. The responsibility is often mission related. So what is the next step is often a question that comes up all the time. We essentially have a race against time. So we are not, we can't drill here until we have reached our goals. Unfortunately, we have so much time, and within that time we have to reach our goal. So and we have to, along the way, we have to make compromises. There might be an unpredicted situation during drilling where it takes longer, the formation is harder than we expected, so it's slower. So then we have to drop another task for that in exchange. So to still be able to, in a timetable, to reach all these objectives that we have. And so we constantly have to weigh what's more important at this point. And so we have two of the major objectives that we have in our mind that we really want to achieve is we want to study the coal in detail, and we want to get the world record of the deepest microbes.
Dedicate with some others.
And now it's already in the hands of microbiologists.
I'm in charge of monitoring the contamination in the sediment cores during drilling. And we do this by adding a chemical tracer compound to the drilling fluid. And then during the drilling process, this drilling fluid is used to cool the drill, so we don't heat things up too much and destroy the samples. And so when samples are then retrieved back on board, I take samples from the outside of cores and also from the inside of cores, and then measure the chemical tracer concentration in that. And using that, we can then calculate what the contamination is with the drilling fluid. And another thing I do on board is extract DNA from some of these samples. And then together with my colleagues, we are then quantifying the numbers of bacteria and archaea, which are the two main groups of microorganisms that we expect to find here. And we are looking at their numbers and how they change. And this will be combined with other kind of quantitative assays in which we will be able to estimate the cell numbers for different types of microorganisms.
See that it really looks like coal. This is fantastic.
I'm on board here as a logging specialist, and the logging is it's a geophysical logging, in fact, and the geophysical logging is to use a kind of sensor that is going to the borehole to measure the physical property of rock type or fluid or many kinds of characteristics of rock are to be measured by that sensor. And this is the ocean floor. And then we start drilling at this point and then go deeper, penetrating to further deep. This is 1,000 meters deep from the ocean floor. And then we are now nearly to 2,000 meters deep from the ocean floor. We are now drilling in this interval, which is marked by that black color. And this black color is the primary target of this expedition because this depth has many reflection seismology feature here. And that indicate there must be many core layers here.
At this point, we just verified yesterday the presence of the coal bed. We only suspected that the coal bed would be here based on seismic evidence, but that's never clear-cut, the interpretation. So now we know it's here, and now we are, in the next 3 weeks, we will find out what it's doing.
My speciality is looking at the organic compounds that you find in rocks. So rock looks like this. I'm interested in this goo that we take out of the rocks. And the thing that I'm doing on the ship is instead of just looking at this goo as goo, I am trying to analyze specific chemicals or molecules within within the rocks that we find here. And the aim of this expedition, one of the key aims, is sort of tracking of a point at which something biological changes into something geological. So coal is made from trees. Trees are biological. Trees die and make soil, and then the soil slowly changes into a rock. And as that happens, the chemicals inside change, and they go from living things to being chemical fossils, and I'm studying chemical fossils.
Do you have a rough idea of the concentrations of the sterols compared to the sterines and steranes?
There are stacks of sterines and steranes, and I think they're probably
more
much
abundant.
more abundant. The main application would be just to explore and understand what's going on. There's been several appraisals of how much biomass or living organic matter is buried in the sediments in the oceans. And this is a chance to not look at it spatially over a map. This is a chance to look at it with depth and see what happens at depth. And it might be there are some levels and some instances where you have a very prolific biosphere, and then suddenly it could be dead, and then suddenly it could spring to life again with the right conditions. What we've been seeing so far is an unexpectedly high amount of chemical fossils that are not quite fully chemical fossils yet, but then again they're not quite biological molecules. So the hope is that if there is a deep biosphere, it should, or it could leave a very sort of clear, distinctive chemical fingerprint that my colleague Yushi could detect.
We are now scraping a core we got which contains a large piece of coal. And the idea is because the coal oxidizes quickly, so we want to scrape the surface inside the glove box. So Marshall is now using a ceramic knife to scrape the outer part of the coal. Once we clean the surface of the coal, because the surface is contaminated by drill mud, we will, some of the code, we will use it directly for our potato experiments. I'm generally very interested in the big question of biodegradation. So, for example, for human beings, we know that we eat apple, we eat hamburger, and this is very clear because we can see that. However, for microorganisms, it's very, very unclear what they are doing and if the organic matter is degradable for them. So we know that marine sediments is one of the largest carbon pool on Earth. So there's actually a lot of carbon down there. However, previous studies, they basically focus on shallower sediment, maybe the upper 1 meter, and then found that actually the quality of organic matter decreased very quickly. So which means probably somewhere 1 meter below the seafloor, the organic matter becomes very, very difficult to degrade. Yeah. But however, the sea is still full of methane, and much of it, based on isotopic value, are biogenic. And also we know that in the sediments, usually you see the alkalinity, which is, you can think of it as a CO2. So CO2 increase, which means they are doing something. They are degrading something. Yeah. So therefore, my general interest is to know if, on the one hand, which microbe is capable of degrade these very recalcitrant carbon. Yeah, that's a very interesting question for me. And we know that coal is made of carbon, basically. Yeah, and it's a very organic, rich stuff. Therefore, I think it's very interesting for me to see if microbes, they can really degrade these macromolecules, because coal is made of macromolecules. Well, for the others, we will use this tungsten mortar to crush it into powder, and then do other incubation experiments. And that's what our goal today. And on the other hand, I'm also interested in knowing if the microbes, they are really degrading something. How do we know that? For example, when you see an apple and you found that there's some crack on it or some strange imprint from the teeth, then you know that somebody has eaten it, right? But how do we know that when we look at the rock, how do we know that the microbes, they have degraded the organic matter they are in? Yeah, so how do I use some molecular approach to see, okay, this is a biodegradation signal. I have been trying to set up some incubation experiments, but these incubation experiments, I use some spatial chemicals, or we call it substrates. And these are spatial substrates that are isotope labeled, so which means they are not present in nature. And the reason to use these isotope label substrates is to see, for example, if you eat a hamburger, then into your body will not record it, because this hamburger is like a sort of like a made from natural product. However, like I know that in a hospital, sometimes if they want to detect some symptom, they will ask the patient to take some C13, so it's a stable isotope, C13 labeled compound. And then later on, they can trace the movement of this compound in the body. So that's the same idea of what we are doing to the microbes. So because it's sometimes very difficult to tell what the microbes are doing because it's not like human beings. I saw you're looking at me, but we don't know what the microbes, what they are eating. So we add these isotope label substrates into the core we took. And then later on, we will do several different things. But in our laboratory in Bremen, what we are going to do is to analyze the lipid biomarker because every cell, they have cell membrane, and membrane is made of lipids. So, so we are going to measure the isotopic value of these membrane lipid molecules. And if the microbes, they eat up the hamburger, for example, and then they use the carbon from the hamburger to build the membrane, then we will see, okay, they are eating up these special substrates.
Just on the level of technology, for us it's not really the challenge to find life, it's more like to find life at this very, very low level in front of the background of all the fossil signals of life, because the sediment holds a lot of signals from past life over millions of years of fossil life, and that is where we see all the molecular signals, and we have to distinguish the few signals that represent actually current life from this large background. That's often a big challenge for us.
Hey,
What's the
Kai.
methane doing today?
Oh, it's doing very well. We had good recovery and get constant readings for the isotope values, and right now
Biogenic?
it's still biogenic, yes. It hasn't changed a lot since we went through the coal bed.
This kind of mission is also associated with risk, with environmental hazards. So drilling into a potential hydrocarbon reservoir is by definition potentially dangerous. So we take precautions. We have that's why we need this ship to be as safe as possible. We have a blowout preventer, and the entire staff is also sensitized given the recent accident in the Gulf of Mexico. So I think the safety regulations are extremely good. That's how I would qualify. But and when we look at the situation right now, from our data that we have, there's not a lot of gas. The level of gas is not that we are drilling into an active petroleum system. I'm really happy about this profile, especially how you can see how it extends the real measured values.
So we are now seeing here the gas coming up from 3,240 meters.
It is very difficult but technically possible to see the deep subsea floor microbes. We have been prepared for the detecting such microbes in deep subsea floor, developing new techniques so that we can discriminate the microbial cells and sediments. So by keeping the cleanest possible condition, I'm assuming that we can be detecting a very deep subsea floor microbes, even in the population is very low. So and by having that, we can be extending the current known extent of the microbes can be lived in the how deep the subsea floor microbes can be there. So by having that information, we can be close to the real picture of the microbial world spread in subsea floor biosphere. There is several limitations can be thought as limits of life. One is a temperature, one another is a pressure, one another is a nutrient limitation. One very strong limit is a space for microbes. We are getting the very hard rock-like geological formation recovered. So there is a very little space for the microbes. So in a case of that, they cannot have the enough space to live on.
One of our big issues is certainly distinguishing dead and alive. And I think this is sort of a gray zone that we are working in. What is really alive, what is dead? Microbes that seem to persist, that hang around for thousands of years, are not really compatible with our standard definition of life. So, and it's hard to measure life. I mean, typically we look for products of metabolism, and if you look on a single cell level, that's very, very difficult because the microbe is so slow. You have to observe for a long, long time.
If you want to know how active a microbe is, then you need to measure the rate with which it takes the nutrients up and produces some products. So, and to measure this, the first thing is we need very fresh sediments, and it should be taken immediately after we retrieve it from the depth, and the microbes should be alive, because if we want them to make the metabolic processes, then they have to be alive, of course. So we have to take care that they are still alive when we start the experiments. So we have to be very quick because all the conditions change when we bring them up. So we prepare small subsamples in unoxic conditions so that they can survive. And then we have to be very quick to start the experiments.
This is a part of a core which is in a depth of approximately 1,630 meters. And what you can see here are very nice structures, sedimentary structures. The first one is here. This light gray thing, this is a burrow. So this is a eating burrow from an ancient animal which is eating up the sediment and fill it again with sand. It's in a brown layer, mainly silt and shale. And very interesting is here this dark black horizon, approximately 2 to 3 centimeters thick, where you have many plant remains inside, which maybe come from an ancient forest, and the plant remains swapped into the marine environment.
I think one of the interesting features is microbes may be contributing the carbon cycles. Using a deeply buried organic matter, they contribute to the degradation of some stuff, and then ecosystem function produce methane at the end. I think such a biogeochemical processes is very, very important, but still remain unknown. Largely unknown. So I think if we understand well such a biological contribution in the Earth's environment, maybe we can figure out some, the nice picture in the future how we can use and save the Earth's health and also sustain our human activities and save the environment.
been in at least 5 research cruises. And normally, in other research cruises, it's very often that scientists, they focus on their own stuff. And of course, from time to time, there is some discussion or exchange of information, but it was not as thorough as like you have seen yesterday, like in the midnight. Each group present their most recent advances. So therefore, we have a better overview of what kind of habitats it is down there for the microbes. Yeah, while in the other cruise, it was like maybe the sedimentologists find something or the inorganic geochemists find something, but the exchange of information is very limited. Therefore, quite often you always need to, after the cruise, you always need to ask that specific person what you have found. However, when you do that only after the cruise, you probably miss the point of making the right decision. That happens very easily.
The scientists, they all work in their own laboratories, and it's the role of us as chief scientists, so my colleague Fumio Inagaki and myself, we have to stimulate exchange between these groups. We have to integrate the data. We have to see if there's something in a data set of one particular group that might be relevant to another. So draw the connections and bring the group together.
Wonderful things about the ship is that there is a very qualified staff of technicians working 24 hours a day.
When we think about the world record, a big issue is, can we penetrate that deeply, drill that deeply in the time that we have? That's a major problem. But there are also more technological problems. Will the borehole be stable from a drilling point of view? That's what we scientists can't influence. That's more within the responsibility of the drilling team here. But borehole could become unstable, collapse, and that would be the end. We wouldn't have time to drill another hole, so that's an issue that's a little bit out of our control. But that could also be a problem. A typhoon could be a problem. If a typhoon comes, which is very common this time of the year, in September, then we would potentially have to leave the site, pull out the drilling equipment and go to a safer place for several days. We would lose time and we wouldn't be able to go for the world record. We just made it. We just advanced the world record of scientific ocean drilling by 8.5 meters. It's a fantastic moment. We are all very happy. 2,119.5 meters below sea level. Well done, Fumio.
Well done. Congratulations to us. I think limit of life is a very important target, but also probably important to know origins of life. It's also, I think, one of the big target of the scientific ocean drilling. And to study the origins of life, I think one of the ways is deep drilling, and how Earth's reaction produce, some bioavailable some compound, and also the polymerization is a very essential part. And I think the key to understand the origin of life may be water and carbon. And if you go deep, like upper mantle, or whenever, if you go deep where there is no life, I think, of course, understanding the limits of life extremely important, but also important to understand the prebiotic reactions.
Looking for life.